Hello, everyone. Thank you for being here. I'm going to be talking about my postdoc research that I'm doing at the Florida Museum Herbarium and University of Florida. Together with Lucas Majur, we're studying a new approach using targeted sequence capture for phylogenomics studies across Cactaceae. Cactaceae is a well-known family that has 1755 species, including highly diverse clades in both North America and South America, as you can see in the map to the right that shows the richness of the species across the continent. This family also has a huge variation in morphology, as you can see in those three pictures here that show different examples of species. Cactaceae relationships have been studied since a long time ago, starting with Sanger sequencing and ending currently with chloroplast genome studies. This is an example of a chronogram that we're building on BEAST using whole chloroplast genome. And in this case, I want to show you how the high support values all across the tree are shown in lighter color like blue, and the dark nodes represent low support values. And as you can see, overall is well supported with the exception of some places in the topology. These tree recovered Cactaceae origin around 28 million years ago. And I'm going to show you some examples of interesting nodes like Leeuwenbergian Pereschia clade around here is recovered as sister of the rest of Cactaceae. But in previous studies, this position has changed. So we don't know exactly if this is accurate. And sometimes it falls inside other clades inside Cactaceae. Next, Opuntii is a huge clade inside Cactaceae. As you can see, it has the shortest branches across all the other main clades, and it has a low support values on the tips. Close related species don't have well-supported relationships. And with this, we know that a new approach to solve the relationship within Cactaceae is necessary, because even including whole genomes of the chloroplast, we cannot solve all the relationships. And we decide to use target capture for this. We use Janssen and collaborators and Johnson and collaborators approaches, first using marker miner with Arabidosis annotated genome as a reference, and then including transcriptome assemblies extracted from GenBank, always trying to represent all the main clades in Cactaceae. Some pictures that show some of those clades, together with some outgroups like Portulaca oleracea. We proceeded to the manual sorting and filtering. This because these loci needed to be hand trimmed to be able to adapt or fit the sequencing technologies that we currently have. And the next step consisted in the blasting process to make sure that we didn't include repeated sequences for the angiosperm 353 in the marker miner set, and also that we didn't include chloroplast and mitochondrial genes. With that, we have a unique lossy, single copy lossy from marker miner that we combine with the angiosperm 353 set to have a final set. The next step consisted in testing these genes with high Piper, and in this case we use 36 transcriptomes sequences, again taken from GenBank, to test this. We were able to recover exonic regions, and this was a successful test of the efficacy of these sequences. We proceed to sequence 96 samples to be able to test the recovery of exon, introns, and supercontigs in a more extensive way. We extracted the DNA in the lab and proceed to sequence a whole plate for this purpose. And I'm going to show you some of the results from this. Marker miner probe set results show overall a pattern that shows a recovery of the genes here with the transcriptome, the 36 transcriptomes here, of high success in some cases and lower in others. So overall, a patchy pattern. We try to recover 122 single copy loci with a size range of 123 to 3,000 base pairs. And we can see in odd cases, we succeed. For example, Stetsonia corine performed poorly, Opuntia coccinellifera performed the worst in all transcriptomes, Ilocereus undatus, Echinocereus pectinatus, and Copiapoa desertorum were additional samples that performed poorly. All this just to have in mind that if some clades in Cactaceae, these probes could perform worse than in others. But when we use this probe set to study these sequenced samples, the 96 ones that we prepare, we see a different pattern, way more dark values, meaning that there was more success in the sequence and way less white. We see that the genes map varied between 107 and 122 per sample. Of course, there were some samples that failed in the mapping, like Phalaenopsis frutescens, Selenicereus triangularis, that had originally very low quantity of DNA, and Cereus fernambucensis. When we tried the angiosperm 353 in the transcriptomes, we have to remember that they were 336 transcriptomes, and they had way more genes. 296 genes were mapped, almost the double than the marker binder probe set, and the size varied between 120 to 2,300 base pairs. And you can see a similar pattern of patchiness where some genes didn't map at all to the samples or very little, and some samples performed very poorly. The same samples that perform poorly in the marker minor probe set analysis perform bad here as well, with the addition of Carnegie gigantea. When we test these probe sets to the sequenced plate, the 96 samples, we saw a huge variation of success in the mapping of the sequences. Between 68 to 296 genes were mapped per sample. As you can see here, there's way more patchiness, more white. So there were genes that perform really poorly compared to others. And the same species or the same samples perform poorly compared to the marker minor, with the addition of 3 samples, Opuntia chlorotica, Melocactus bellavistensis, and Opuntia tapona. And in this case, the last 2 probably were due to samples with little DNA originally. And finally, I'm going to show you some results of the preliminary phylogenies that we are building. This is just an example of our XML analysis that we run using the concatenated supercontigs with a strict filter, allowing very little missing values in both assemblies. This is marker minor, this is angiosperm 353, and the nodes are tied or linked by these lines, and the color is just arbitrary, and the similar the color, the closer the relationship between the tips. Here you can see that there are differences, but the main one that I want to point is the Leeuwenbergian Pereschia clade, here marked on red. On the marker minor set, you can see that is inside another clade, is sister to Cactoideae, while in Angiosperm 353, it's sister to the rest of Cactaceae, in a similar way that we observed in the chloroplast genome tree. So there are differences between the sets depending on how you use it. And the same here, marker minor versus Angiosperm 353. These are support values shown in the tree. The lighter the color, the higher the support. The darker the color, the lowest the support. And you can see, hopefully, just by comparison, that marker minor tend to have lower support nodes in the medium and deep levels. And Angiosperm 353 shows low supports in very shallow levels overall. Of course, there are exceptions, but I think we observe a trend in these two probe sets, and it's worth to keep studying or exploring. And with that, I want to just conclude that our results reveal great potential for the combination of a Cactaceae-specific probe, the marker minor set, and Angiosperm 353 probe set. And not only the combination of these two, but the study of these each separately with Cactaceae and other clades inside Cactaceae. And with that, I want to thank you all for coming, and I'll leave my email here if you have any questions or ideas of collaboration. We are very excited of further studies with this probe set in the family. and I'll take any questions from the chat.